Okay, now we focus on physical hydrogels, which we said are those hydrogels that are cross-linked through secondary interactions, reversible interactions, or non-covalent interactions. Those could include those that are held by electrostatic attractions between oppositely charged polymer chains. We could have hydrogels that are cross-linked through polymer crystallites. We could have hydrogels that are cross-linked through affinity complexes, meaning molecules that have a high affinity toward one another. We could have cross-links that are made through supramolecular assemblies, hydrogen bonding, hydrophobic interactions, or just physical entanglement. As we mentioned, typically these type of hydrogels are not homogeneous because you would have typically clusters of entanglements or regions that are hydrophobic and crystalline versus amorphous regions. And the crosslinks are typically going to be I should say the crosslinks, yeah, are typically going to be clusters that are bringing together the chains. So you would end up with a non-homogeneous system. Now, importantly, the association in these physical hydrogels is reversible, meaning that we can disrupt those crosslinks through changing some of the conditions that are surrounding that material. For example, changing the pH, the temperature, the concentration of the polymers themselves, the ionic strength, or the presence or absence of competing solutes, and mechanical stress as well. All right, we're going to start by looking at an example of ionic hydrogels. So an ionic hydrogel would be that which is made by ionic or electrostatic interactions. And there's basically two ways to be able to form these hydrogels. One of them is called ionotropic gelation. In this ionotropic gelation, what happens is that you begin with a polyelectrolyte, either a polyanion as shown here or a polycation. We then interact that polyelectrolyte with a multivalent cation or multivalent ion of the opposite charge, I should say, and that ion is basically going to bridge chains together in order to form the hydrogel, right? Another method is called complex coacervation. In complex coacervation, you basically begin with two polyelectrolytes that have the opposite charge. So you would have a polyanion and a polycation. When you bring those two together, they're basically going to cross-link due to the attraction between the opposite charges. That again is called a polyelectrolyte complex or PEC hydrogel. The second type of hydrogels are those that are held by crystalline regions, basically crystalline associations in between polymer chains. Here on the right hand side we have an example. This is for polyvinyl alcohol. Polyvinyl alcohol actually can be made to form crystallites through a couple of different methods, but one of the methods is this freeze-thawing method, where you subject the polyvinyl alcohol solution through several cycles of freezing and thawing, and thawing, to allow the polymer to form these crystalline regions where the chains are stacked with one another and are interacting through hydrogen bonds. Depending on the level of crystallinity of your system, you might end up with a clear system, a polyvinyl PVA hydrogel that is transparent, meaning that you have a low extent of crystallinity. And in this case, most of the hydrogel might be held simply by hydrogen bonds, or you might end up with an opaque hydrogel that indicates that you have a high level of crystallinity, a lot of these different microcrystals that are basically junctions in between a number of chains of the polymer. The next type of hydrogels are those that are made from affinity interactions, meaning that we have molecules that have a high binding affinity for one another. That affinity might be a result of having the right size, the right shape, the right chemical conformation that enables very specific interaction between the target and the affinity molecule. perhaps. There would be, typically there would be hydrogen bonding as well as electrostatic interactions that can bring those two together with high specificity. One of the examples of affinity complexes that are utilized a lot in biomedical materials is this biotin and streptavidin or biotin-avidin interaction. And we see an example of that here on the right-hand side. So biotin is a small molecule. It is also known as vitamin B7, and its structure is shown down here at the bottom. So biotin has very high affinity for either avidin or streptavidin. Avidin is a protein that is produced that can be found in the whites of eggs of many birds and reptiles. And then streptavidin is a similar protein, but that also has affinity for biotin, but that is obtained from a bacteria. Both of these proteins are tetrameric proteins, meaning that they have 4 different regions that have a similar structure, and each of those 4 regions is able to bind one biotin. That's the reason why in this particular graphic they represent streptavidin as a square with 4 different sides. Those 4 different sides can each bind one biotin molecule. So in this particular example, what they did was to start with a polymer that was a polybiotin, meaning that we have a polymer that has been decorated with biotin several places along the chain. And so if you bring this together with streptavidin, the streptavidin would be able to bind to at least 2 different of these biotin molecules that are decorated on the polymer, and would be able to bring polymers together in order to form a hydrogel system, a cross-linked system, where the cross-link is simply the interaction, which is a non-covalent interaction between the biotin and, in this particular case, streptavidin. Again, just to mention, this is a non-covalent interaction. But I will go ahead and mention that the binding of biotin to avidin or to streptavidin is one of the strongest known non-covalent interactions. And so it is utilized a lot in bringing things together, like as a bioconjugate system. You might see biotin and avidin being utilized for a lot of biosensing systems and a lot of immunoreagents to be able to bring together, let's say, an antibody together with a dye or something like that. All right. Other types of affinity complexes that could be utilized would be coil-coil interactions between peptides. And so you might imagine a hydrogel that is cross-linked through the utilization of polypeptides that have affinity to one another and that can coil and form hydrogen bonding between the chains. Similarly, we could utilize oligonucleotide complement interactions, which would be held together by base pairing or hydrogen bonding, basically, but that have high affinity because of the complementarity between the sequences. And then affinity ligand, such as antibody-antigen or aptamer-target interactions. So antibodies are proteins that are made by organisms, including humans and animals, that have specificity and high affinity toward an antigen, which is typically a foreign object or a microorganism. And then aptamers are a similar type of molecule, but that are made out of oligonucleotides. So they serve the same role as an antibody in that they are molecules that have the ability to bind to a small target or to another target, but these are made out of oligonucleotides versus being proteins. I do have an example of the antibody antigen, and that's in a later slide. In this second example of an affinity hydrogel, we're actually utilizing this molecule concanavalin A. This molecule is a lectin, which is a carbohydrate binding protein. And this one has the ability to be able to bind sugars such as mannose and glucose with high affinity. And so in this particular case, its ability to bind to glucose was utilized. Basically, what they did is that they started with concanavalin A. They reacted it with this small molecule. This part of the molecule is N-hydroxysuccinamide ester, which is reactive toward primary amines present on the CON-A. So CON-A, again, is a protein, so it has a number of primary amines. This NSA molecule can bind covalently to those primary amines, and so by utilizing this, they were able to introduce this vinyl group onto the CON-A, and that vinyl group is shown right here. All right, so from there, the next thing that they did is that they utilized this HEMA molecule, or GEMA molecule, I don't know how to say it, or GEMA. Okay, this GEMA molecule is basically a glucose derivative that has another vinyl group right here. That vinyl group, remember, is a CH2C double bond that can be polymerized through free radical polymerization. So that's what they're doing here. Initially, the CON-A is functionalized, kind of getting it ready to be able to be polymerized. Here now they add a glucose molecule that also has those vinyl groups. Remember that the CON-A can bind to glucose, so when we interact these two, what ends up happening is that the CON-A binds to the glucose, and you end up with a complex system. Each of these is the modified glucose, or GEMA. Now you have a complex system that has the ability to be polymerized. So the next thing that they did is that they mixed that complex together with a monomer, diethyl amino ethyl methacrylate. Again, notice that it has that vinyl group right here to be able to enable it to be polymerized. And then they also included polyethylene glycol dimethacrylate, which acts as the cross-linker because it has two of those vinyl groups. When they mixed everything together, they also included ammonium persulfate, which is a free radical initiator, and TMED, which is an accelerant to enable that polymerization to occur at room temperature. And in this particular case, they did it at 36 degrees Celsius, and that basically generates a polymer network system, which in this case was in the form of particles. Here we have an image of what this system would have looked like. Basically, now we have these blue polymer strands, which are made from the diethylamine ethyl methacrylate and the polyethylene glycol dimethacrylate. This one does act as a cross-linker, chemical cross-linker, but in addition to the chemical cross-linker, we also have the CON-A acting as an affinity-based cross-linker in between polymer strands. And by utilizing this, if now we expose that particle to free glucose from, let's say, the physiological fluids of the patient, then that glucose would compete for the binding spots of the CON-A for glucose. Basically, the glucose from the patient could come and compete with these sites and basically free the polymerized GEMA, thereby increasing the porosity of the hydrogel and enabling you could potentially utilize this to enable the release of something like insulin if you had loaded insulin within the polymer network. So this is a really interesting system that could potentially be utilized for the delivery of insulin for diabetic patients. Another example of a physical hydrogel cross-linked through affinity interactions is this system where they're utilizing an antibody and a target or antibody-antigen interaction. I don't recall, to be honest, what the target was in this particular system, but what was interesting is that they basically polymerized the antibody. So antibody is a protein, it's a large protein that again has the affinity to bind to a particular target molecule. That antibody in their case was modified with NSA, which is the same molecule that we had seen in the previous system, which has basically the ability of now forming an antibody that has a vinyl group that enables it to be polymerized. Similarly, they took the antigen and they functionalized it in a similar way so that they could polymerize the antigen as well. The antigen is the molecule that the antibody is able to bind. After they had that, they basically polymerized the modified antibody in the presence of acrylamide and ammonium persulfate as the initiator to now get a polyacrylamide that has antibodies on several parts on that chain. And then separately, they took the modified antigen, they also polymerized it with acrylamide to now form polyacrylamide that is decorated with the antigen. They did this second polymerization in the presence of the first polymer, so you actually ended up with an interpenetrating polymer network, which is what basically they're trying to show here. We have, let me zoom in a little bit. Oops, undo. Sorry, I will get this eventually. All right, I know it's a little pixelated, but hopefully you can see. There are two sets of polymer chains. One is the dark black lines. That would be the polymer chain that has the antigen. And then we have these dashed lines, which is actually the polymer chain that has the antibody. Initially, when the two things are together, notice that the antibody is basically binding to the antigen, which is the little circles, the open circles. And that enables the porosity of the hydrogel to be low and thereby the hydrogel to be in a shrunken state. Whenever now that hydrogel is exposed to soluble antigen, let's say from the patient, physiological fluids, then that soluble antigen would displace or would compete with the polymerized antigen to be able to bind it. And in doing so, it would basically release the polymerized antigen, thereby increasing the porosity of the hydrogel and enabling the hydrogel to swell, which, as we mentioned in the previous example, would enable the hydrogel to release its cargo if we had encapsulated a therapeutic cargo within. All right, using this system, they were able to show that the hydrogel did in fact swell as a function of antigen concentration, and they were able to show that they could do this in cycles, which could potentially be utilized for on-demand drug delivery, let's say for for insulin or something like that that is needed at specific times whenever the concentration of the antigen is high in the physiological fluids of the patient. The next example has to do with the use of supramolecular assemblies. So these supramolecular assemblies are basically transient associations between a molecule that contains a cavity and a molecular guest. So the molecules that have the cavity are those that are shown here on the right-hand side. This includes cyclodextrins, which are natural compounds, and also synthetic compounds, including the other three that we have here. Calixiranes are an example that you might see a lot in the literature as well. But yeah, cyclodextrin is utilized a lot. So what happens is that these molecules, these cavity molecules or cavitants, they have the ability to bind to specific molecules, typically through hydrophobic interactions. So you could imagine that I can use my cavitant and the molecule that is able to interact within, and when they come together, they would form this kind of complex. That interaction between the two could be driven, as I mentioned, through hydrophobic interactions. In certain cases, you would have pi-pi stacking or van der Waals forces. And in some particular cases, you might also have hydrogen bonding or even electrostatic interactions that are bringing them together. But they basically have high affinity for one another and they're fairly stable. You could imagine utilizing this kind of lock and key binding method as a way to cross-link the hydrogel, and that's what's shown here. So basically we could bring individual chains together by enabling the interaction between a polymerized blue molecule and a polymerized cavity molecule. And then the green thing is basically the cargo that would be carried within the hydrogel. In this particular example, what they were showing is that you could actually utilize this to generate an injectable hydrogel. So for example, here we have a hydrogel that has already been formed within a syringe. Then by passing it through the needle, we create enough stresses to be able to break apart that interaction, because remember that these are reversible interactions. They're non-covalent interactions. So just by applying that mechanical stress, we can actually break them apart and enable that hydrogel to be injected. Once it is within the tissue, then there would be a recovery where the bonding or the assembly, the supramolecular assembly would occur again. It would basically lead to the cross-linking of the hydrogel again. And then over time, through competitive interactions with biomolecules, the hydrogel would begin to disassemble, because the binding between the cavitant and the target would kind of be competed away, and the hydrogel would dissolve over time, thereby releasing the cargo over time. So that's a really interesting injectable drug delivery system based on a hydrogel that is held by supramolecular assemblies. All right, so this last slide that I wanted to talk about in this particular video is different methods that could be utilized for the synthesis of physical hydrogels. In this particular table, we're focusing on physical hydrogels. One of the methods is to warm a polymer solution in order to form a gel. So for example, in this particular case, you would be utilizing a polymer that has an LCST behavior, for example. Let me go ahead and put that here. So in this case, that would be a polymer system that has LCST, or lower critical solution temperature ability, which when you warm it up, it actually forms hydrophobic interactions that enable the cross-links to form, right? So for example, we could have a hydrogel that is made from poly-N isopropyl acrylamide, or that has regions of poly-N isopropyl acrylamide, which upon heating above the LCST would generate aggregates which act as the crosslinks. Number two is to cool a polymer solution to form a gel. In this particular case, we're basically talking about forming hydrogels that are cross-linked through hydrogen bonding. Hydrogen bonding could be something like a polyvinyl alcohol gel, or an agarose gel, or a gelatin gel, or even a gel that is cross-linked through DNA interactions, DNA-DNA interactions. Number three is to cross-link a polymer in aqueous solution using freeze-thaw cycles to form polymer microcrystals. And an example of that was the freeze-thaw method for polyvinyl alcohol, which led to the formation of microcrystallites. Number four is to lower the pH and form a hydrogen bonded gel between two different polymers in the same aqueous solution. For example, polyethylene oxide, which is the same thing, by the way, as polyethylene glycol, same chemical structure, PEO and polyacrylic acid. So in this particular case, they say to lower the pH because polyacrylic acid at neutral pH would be deprotonated. And in this particular case, what we want is the carboxylic acids to be in their protonated form, COOH, which is neutral, but which would have the ability to now hydrogen bond to the polyethylene glycol, right? If it is protonated, it's able to hydrogen bond to polyethylene glycol, which we all know has this structure. And sorry, my writing is terrible using this tablet. And again, we would get hydrogen bonding between these two, and that would basically form the hydrogel through physical hydrogen bonding crosslinks. Number five would be to mix solutions of a polyanion and a polycation to form a complex coacervate gel. For example, utilizing sodium alginate plus poly-L-lysine. Sodium alginate can also be called alginic acid, and so it's negatively charged. And polylysine has primary amines, which would be positively charged at neutral pH. So basically we have a polyanion here, polycation, and the opposite charges bring them together into forming a complex coacervate. And number six is to gel a polyelectrolyte solution with a multivalent ion of opposite charge. For example, taking sodium alginate and cross-linking it with calcium ions that are divalent, that would be able to bridge two different polymer chains. Another example of this last one is actually the one that we discussed in the presentation by Ashley, if I remember correctly. It was a carboxylated agarose that was cross-linked with zinc. The zinc ions, the zinc 2 plus, basically would be able to act as that cross-linker. All right, that is it for physical hydrogels. See you on the next video.